Faccio io? Okay. Okay. We have now the next topic, which is called, which is based about latent genetic configuration associated with Alzheimer's disease. And I ask Professor Annalena Venneri to introduce the speaker.
First of all, it is a great pleasure to actually be invited to introduce Dr. Lia Talozzi. The story of Lia Talozzi and I's encounters is slightly different from the one described for Francesco and Professor Miragoli. I didn't know Lia. Lia is not my former student because, of course, I had been in Italian universities only for a little while. And Lia actually, I think this is the wrong slide, yeah, isn't it? No, it's not the beginning. It's the end, actually.
With the toxicity.
Yeah.
Or
It works.
Point,
That's okay, don't worry, I can use this one. So what I was saying is I didn't know Lia because Lia is actually not somebody who has studied in Parma. She has a degree in physics from University of Bologna, and she also did a PhD in biomedical and neuromotor sciences, always at the University of Bologna, where she also became an MRI specialist. But of course, in my research, I do have a lot to do with MRI, and especially the interest of Lia was in Alzheimer's disease. So we did meet at one of the main conferences of the year, at the Alzheimer's Association International Conference. It was in the United States. Lia approached me and we discussed research, not specifically related to any possible future working together, but just simply the fact that we had shared interests. But then she applied to another university for a Marie Curie fellowship. She received the Seal of Excellence, and at that point, knowing that I had actually moved to Parma, she came to ask whether I was interested in being her mentor for potentially applying for next generation EU funding. And that's what actually happened. Lia, I was very pleased because here Lia had those skills that were missing in my lab. She had been in excellent labs in the United Kingdom as well as the US. She was, during her PhD, she spent some time in a prominent university in London, at King's College, where she she took the opportunity during her PhD to actually acquire skills in, especially in the diffusion tensor imaging. Then she moved to Bordeaux for a postdoctoral fellowship, and she worked with one of the world specialists of tractography and DTI imaging. She worked on the human condensome, and she worked on strokes primarily, but her skills were transferable to those things which I'm interested in. Then she moved to Stanford for another postdoctoral fellowship, and there she moved to a different field, that of genetic of Alzheimer's disease. And that brings me to the focus of her research, which is Alzheimer's disease and neurovascular disorders. These are not so distant because often what we see in Alzheimer's disease is vascular comorbidity. And the fact that she had an interest and skills in that particular field convinced me that she was a good person to invest in and to sponsor for her application so that she could join our team here in Parma. Lia has a range of skills, not only in neuroimaging, but also in neurogenetics, and especially in computation models. She's been a fantastic addition to our team. The work that she's been doing since she joined Parma is the focus of her talk today, where she will be looking at profiles, genetic profiles in Alzheimer's disease. Now, for those of you who don't know what Alzheimer's disease is, it's the major cause of neurodegenerative dementia. And although it is a disease that affects, the later part of life, it is a significant burden to national health services and families. With know that the majority of this disease is caused by, sporadic, is a sporadic condition, doesn't have a strong genetic signature. However, the latest research in this field tells us that although, we don't have a gene causing this particular form of neurodegeneration, there is a profile, either in terms of polygenic risk or in other aspects of genetics of this disease that actually may be useful to study. It might be actually a good signature that might help identify the disease earlier. And that brings me to the end of Leah's presentation, and I invite you to come and give your talk.
Thank you, Professor Venneri, for the introduction. Yes, it's true that sometimes like you cross people to conferences and then you can end up working together, and that's amazing. That's really one of the magic of science. And yes, as we discussed a bit in the round tables, I applied for a global fellowship. So actually, my project, my Marie Curie project, started when I was in Bordeaux during my first postdoc. And the idea was to move to Stanford, where I was going to do the outgoing phase of my global fellowship. But then, as you know, we received the Seal of Excellence, and I didn't receive that findings, but I got a call from Stanford that they say, hey, but if you want, you can come and work with us. But we are working mostly on genetics. We know that you are doing imaging, but if you are interested, you can expand your research vision field. And so I started also to work in neurodegeneration and genetics. And now I moved back, and so I took this project and kind of we are adapting it to the new cohorts that we have with in the Venneri lab, and thanks to the foundings of the Next Generation AU here in Parma. So I will talk about Alzheimer's disease because it's a really complex disease. We saw heart failure that has a great complexity, a lot of presentations, and also there is a bit of genetics, the macroarrays, and so also Alzheimer as its entire world. And also I'm going to show some preliminary results that we are conducting here in Parma. These are from the European Prevention of Alzheimer's Disease cohort. This is a really interesting cohort because it's looking through prodromal stages of Alzheimer's disease, so really at the beginning of this neurodegeneration process, where you can even have no symptoms at the beginning, but your biomarkers can be different for like a normal aging profile. So as I was saying, Alzheimer is really multifactorial, and I'm going to try to explain you a bit. You have an initial biological phase, and so you can really look at the protein levels. You can see how the amyloid precursor protein, the APP, is processed, and then it will generate in some peptides, the amyloid beta peptides, that if are not cut and formed in a correct way, they can aggregate. And these are forming plaques. And these plaques are kind of toxic for neurons. And so all the environment is a bit compromised. And also you can have tau accumulations. These are forming tangles inside the neurons this time. And also this is not good for neurons because over time it can bring to a neuronal loss. Then this is really the first biological phase. Then you have a kind of secondary cellular dysfunction. And there are a lot of questions, which is the time course of all these issues, but I'm trying to give you like a general idea. And so you can have some issues in the neurovascular unit that can also bring to a dysfunction in the cleavage of these proteins and the alterations of the APP metabolism. And this all seen in the context of the nervous system. Also the microglia can contribute to these dysfunctions with like an immune reactions, the astrocytes, the oligodendrocytes, with the lipid metabolism. So everything started to not work correctly. And so this can bring ultimately to neuronal loss and all the neurodegenerative process. And coming then to MRI, as a physicist, I usually go to the scanner, acquire patients, tuning the weights of MRI, because you can have many information from a single scan. And here you can see the structural MRI, so you can clearly see the white matter inside that is kind of lighter, and then the gray matters, and when there is neurodegeneration, you have the loss of gray matters, and so the liquid inside the brain, it's kind of expanding, and these are the ventricles. And ultimately, then you have clinical symptoms. Here in Parma, we are working in a lab with a lot of psychologists that really test the patients. They measure the memory loss at first, but also other dysfunctions like the executive, language deficits, visospatial deficits, and also you can also have some behavioral changes. So this is kind of the big picture of neurodegeneration, and then you can see all of these under the lens of genetics, because Alzheimer's disease, it's also hereditable. We don't know much about this in the case of sporadic dementia, but there are genetic risk factors that, thanks to massive multi-cohort analysis, we now have the statistical power to see them, and here I reported some of the genetic factors that I'm going to talk in a second. Going back to neurovascular, so in part of my research field, research path, I work on stroke, and there you have like a clear neurovascular damage because you have a lesion, and that is really clearly detected in the brain. In neurodegeneration, the process is a bit more kind of hidden, so you can have many kind of neurovascular deficits. You can have like more inner lacunes, you can have hypoperfusion, you can have like dysfunction in the cerebral small vessels. I want to point here the small vessel diseases, and all of these dysfunction can ultimately bring to atrophy, and so that was showing before with MRI, so the loss here of the gray matter and the liquid here in dark, that is kind of the sign of atrophy. With the genetics, we can really kind of classify, let's say, participants if they have a mutation or no. In the case, so here is the DNA, you can have a single mutation of a nucleotide that is called SNP, the single nucleotide polymorphism or variation, SNV. So you can have like a mutation, a different letter from the most common presentation. This alteration can actually bring to a change in the amino acid, here from a cysteine to an arginine, and this really a change of one letter and one amino acid can bring to a different protein isoform, and this isoform can actually not work correctly and increase the risk of Alzheimer's disease. This is what has been shown in the highest risk factor for Alzheimer's, mutation in the apoprotein E. So here you have the ApoE2 high variation, let's say, that is actually protective for Alzheimer's disease, ApoE3 that is kind of neutral, and the ApoE4 that is actually risk increasing. And here you can see that you have these two arginine here, and the shape of the protein kind of change if you confront it with ApoE3 here and ApoE2. And this changing really impact all the process of, for example, lipids or the aggregation of amyloid in your brain. So these are the, so here you have two axes. In the x-axis, you have the frequency in the population. So you're gonna have like rare mutations, more common, up to 15%, let's say, and like in a medium common, let's say, and then more common mutations. And here you have ApoE4, so the risk factor for Alzheimer's disease, and you can have it in one copy, because your DNA you hereditate from your mother and your father, so let's say one copy of your DNA, and then homozygous, two copy, that is, so, and here the risk for Alzheimer's disease is increasing. You can also have some mutations that are really impacting the APP metabolism, so on the APP gene, but also the PSN1 and PSN2. These are really, really high risk factors, and if you have these mutations, they say that are really penetrant in the familiar pedigrees, but the good news is that are really also rare, so below the 1% of the populations are carrying these mutations. And also you have mutation in other genes that are kind of touching other like biological function, so the tau metabolism, the cholesterol, immune response, the endocytosis. And so just to have like a complete view, here I'm gonna show also the common mutation in these other genes. So Alzheimer's disease is really multifactorial, but also from a genetic standpoint, it's really complex to understand which are the players that can either protect or increase your risk for Alzheimer's disease. Going back to kind of like a clinic view that I like, how do we measure these alterations that we have in neurodegeneration? You can measure the accumulation of amyloid and the phosphorylated tau directly in the brain, in the CSF, and also in the plasma. And over time, these biomarkers really established, starting from the CSF, that is kind of the direct measure, the amyloid PET, but also the tau PET. And now we are really value also blood biomarkers that are kind of less invasive than the other methodologies. So what is happening in the CSF? If you have an increased level for phosphorylated tau, this is pathological, so is indicating you the presence of Alzheimer's disease alterations. On the other way, if you have decreased amyloid beta, this also is meaning that you are kind of a D positive, because we don't really know why this is happening, but the general idea is that if the amyloid is accumulated in plaques, is less detective peripherically, for example, in the blood. And in PET, you directly measure the presence of the proteins. So in this case, if the tracer is measuring amyloid and tau, if they're high, they are like a D positive biomarkers. And then in the plasma, that is mimic the behavior of the CSF. And so also there we can measure the amyloid and tau levels. This is the MRI scanner here in Parma that we are using, and we are setting to kind of, now we are working on a protocol that is always in the neurodegenerative field of research, but it's looking to patients that have mild cognitive impairment. So again, a prodromal stage of Alzheimer's where the symptoms are not so evident and severe as in Alzheimer's disease patients. This is a 3 Tesla scanner, and so here the patients will lay on this bed, and people generally ask if the scanner is closed and open. Here you can see that just the brain, the head goes inside the MRI scanner, so it's kind of like a comfortable setting, let's say. And from MRI you can have many measures, and for measures I mean different contrast in imaging. Here is the classical T1 weighted image where you really measure the morphology of the brain. And here I just pointing where we have a neuronal loss and so an atrophy. In this case was a patient with mild cognitive impairment, so even if the symptoms clinically are not really severe, you can start to see some alterations in the brain scans. Then you can also measure those effects of neurovascular damages, and you will see it here as upper intensities in the FLAIR T2 weighted scans. And these lesions can, they will not classify a patient that have Alzheimer or not, but they can give you they can give us information of the neurovascular fit health of our brain. So if you have these hyperintensities, probably there is a bit of sufferance in those areas. And as I was saying, the neurodegeneration process is really a long process. You can even have a positivity to biomarkers. The first one to be positive is the amyloid beta accumulation, even in cognitively normal people. Then moving to the mild cognitive impairment phase, where some of the memory symptoms, so I could say like here in red you have the amyloid beta, then the tau in blue, the brain structure, so the atrophy I was showing here in green, the memory deficits, that is the primary deficits that usually appears in Alzheimer's patients, and other clinical dysfunctions. So in mild cognitive impairments, all these biomarkers and clinical measures, they started to go up, but only in the dementia stage you have really positive measures for all these biomarkers and clinical measures. But we are interested in see what is happening here, if we can really see something in advice that help us in understanding the trajectories of patients, and also like help them in going through this process, and hopefully even stop the neurodegenerative process. And so I'm showing here some of the results of a study that we actually submitted to the Alzheimer Association conference, the one where we met in Philadelphia with Professor Venneri, but this time it's going to be in London. Actually, these analyses have been conducted primarily by Karen, one of the trainees in my lab that I have the pleasure to kind of mentor in her thesis. This is a really interesting collaboration because she's doing a master in AI, and so we are really leveraging this big data set to find new measures, new patterns that haven't seen before. And so we took the data from the European Prevention Alzheimer's Dementia cohort. So this is a big study, 21 sites, but with a for example, MRI scans were acquired with a really harmonized and good data set that is not so, that doesn't happen often in the clinics where all the measures are different. Here there was a vision, and so the cohort is really well characterized for Alzheimer. They acquired over 1,500 participants, and these participants have, most of them have history of dementia in their families. So we're gonna see that we started to see some of the biomarker changes, even if they are cognitive normal. And then they acquired really a large set of multimodal imaging data, the 3D T1 weighted image that I showed you before for the neuronal loss, the FLAIR for the vascular deficits, but also ASL, diffusion, and resting state that are more advanced measures. But here to start, we are looking to the FLAIRS dataset to see if we can gain some information from the neurovascular damages in the brain. So we analyzed 1,720 scans at the baseline because it's a longitudinal data set, but we start from the baseline. They are relatively young for a neurodegenerative cohort, in average 65 years old. They are 44% males. And if we look to the genotypes of APOE2 and APOE4, we can notice that it's kind of different from the general population, where the APOE4 variant is 15%. That was the risk increasing variant for Alzheimer's disease. Here is 21%. So we kind of start to see some of the family history there. And then the protective area on the other way around, the E2 is lower, 6% against 8% in the normal population. And also, if we look to the range of amyloid beta and phosphorylated tau measures, even if they are on average cognitively normal, and you can see it here from these two clinical measures, the mini-mental, that is on average 29, the maximum is 30, so they are okay. And the CDR, the clinical dementia rating, that it's 0, it's cognitive normal, and 1 is start to be impaired, 0.5 is mild cognitive impairment. And so we are interested to correlate our FLAIRS measures to these variations of biomarkers, even if the clinical presentations are not severe now. We are at the beginning of the slope that I was showing before. So we start from the segmentations of these hyperintensities in the FLAIR scan, and then we do it for all the data set. And this is a group variability map. So you're gonna see in yellow where most of the participants have lesions, the 94%. This is usually here close to the ventricles and in the optic radiation that really have like a high metabolism. But you can see then in red that there is a great heterogeneity of lesions all over the white matters, that they're going to be different from a participant to another. And to measure this variability, we need a high-order modeling, and we used dimensionality reduction. So putting this information, and we are talking about thousands and thousands of boxes and intensities, into spatial components, so axes where we can actually see the patients like a point, and I'm going to show you in a bit. We used a dimensionality reduction that was the Laplacian components, and we actually use also others like principal components, but I'm showing just here the best results. So here you have the first component and the second component, and each dot in the graph is a subject, and you can see how the spreading of the subject also started to create kind of two clusters here, and you are showing, like measuring a variability, modeling the variability. And then you have also the other components, and we evaluated up to 7 components. And here you can see how the distribution of subjects is different according to the different components that are mostly independent, and they are give you different informations about the lesion distribution. So we correlated this with the biomarkers, the plasma, in the plasma, the amyloid and tau levels, and also the clinical severity, the CDR and the mini-mental, and we corrected in our model for also age, sex, and the genetic ApoE variants. And I'm showing here the best results that we got because there are a lot. Here for the CDR, the clinical dementia rate, we got the best results with a p-value of about minus 0.9. And you can see here that with a CDR of 0.5, you start to have some impairment, let's say mild cognitive impairment, and 0, you are healthily cognitive, so no issues. And you can see here in the distributions doing kind of this cutoff that are different, and they actually follow a shape of the first Laplacian component. And you see this also for the amyloid beta. So generally here 1000 is the cutoff of the amyloid beta positivity. Below this cutoff is positive, and above is negative. And here again we can appreciate a significant difference, and this time the information so from the variability is coming from the second Laplacian component. And then also for the phosphorylated tau, the positive cutoff here is 27. So above this cutoff, they are positive, and below negative. And again, we have a different distribution, but the information comes from a different component, so the component number 7. So really, we are measuring a signal, a difference, informative of biomarker status, but starting from the neurovascular damages. And how we were able to do it, doing this dimensionality reduction, and specifically the Laplacian embedding. Key findings, I don't know how I'm doing with time, but I'm gonna conclude. And so we saw that white matter hyperintensities are actually informative of clinical presentations in the setting of Alzheimer's disease, as the CDR, and also the neurodegenerative biomarkers as amyloid and tau. And we actually saw that the specific Laplacian components correlate with specific biomarkers, so was really helpful to disentangle, to do the embedding of the variability, because sometimes white matter hyperintensities are overlooked because are kind of complex and too heterogeneous to study. And we see this as a really important information in the early stage of the pathology. And so we hope to kind of find a new algorithm, a new measure to disentangle the complexity of white matter hyperintensities. And here I would like to thank my mentor here in Parma, Annalena Venneri, in Bordeaux, Michel Timboud de Scotland, where I did my first postdoc, and Michael Gratian in Stanford, where I was there as a postdoc and then junior faculties. They, both of them helped me in my Marie Curie writing. My lab members, Riccardo Manca and Michaela Mitolo, that are psychologists here, that they really follow patients and give me a lot of insights about the neuropsychologist. And also Karen Moreale, the trainee in our lab, that she's doing an amazing job with this data set. So that's all. Thank you for the attention.
Any question? Because I have two.
Okay.
The first one is just for you. The second one is for both. Okay. So the first one is your Master of Science is in physics, and then you got a PhD in neuroscience. So you got close to biology kind of later in time.
Yeah.
Was this good? Was this bad? Was this helpful in the way you approach science?
So I would say that I was always close to, in my interest, like generally in biology and medicine.
And you went for physics.
So it was, no, I started from classical studies, so humanities. Then I wanted to go to medicine, but I didn't know anything about science, basically, because I just studied Latin and Greek. And so then I say, okay, let's do physics, because I really like actually relative theory. And then after 3 years of physics, I say, okay, but I actually like biology. I know that I have like a kind of mathematical mind, but so I do coding, all that stuff. But my interest was actually, like, I always be interested in neuroscience, and that interest actually like sparked by chance, because I bought a book that I thought that was about philosophy, The Error of Cartesio, but it was a book about neuroscience written by Damasio, where every chapter was a clinical case. So starting from there, I say, like, maybe I like neuroscience. And then there was this master in Bologna that is called Applied Physics. So you do all the physics that is behind the MRI, the PET, and then I think I literally fell in love with imaging. So for me it was amazing, this idea to understand how a 3D image was created. And then I started to work, I did an internship in the hospital, and there I started to work with radiologists, psychologists, neurologists, because I have this interest of neurology. And then I did the PhD in the biomedical field. That it's kind of a broader, but it was neuroscience.
Okay, that's great. In Italy, we are not really good at mixing things up. We keep our path even in when you apply for a project. We were discussing, I don't remember with whom, outside. If you apply for a project and you know that there's going to be a panel that will evaluate your project, you need to make sure that in the panel there are people that studied what you studied, that has the same degree that you have. And so this is actually a limitation. So we all fall into settori disciplinari.
For me, no. I've been in all the sectors.
No, exactly. But that's, I think,
psychology.
this is how it should be. So we need to change. You're a good example. And the second question is, okay. Well, the second one is very specific, and it's for both. And it's you both talk about chronic diseases. And you think about therapies understanding them, and in your case also trying to kind of correct the problems. And both neurodegenerative diseases and cardiovascular diseases have a link with your choices. And for example, the diet. I wanted to know if you think that the diet, despite a genetic marker, despite a genetic predisposition, and in particular because of the vascular component, do you think the diet can actually still help, or we're all doomed based on our genes to die of one of the two things?
Okay, I will start. So before Francesco said the word that I love, that is pleiotropy. So from the Greek, pleios, like in many tropos, in a different locus, places.
And you see the classical studies
the
here,
classical studies. And and for example, for the APOE4 genetic variant, it can impact, it has an impact of Alzheimer's disease, but also cardiovascular risk factors, and it's linked to the process of cholesterol. So I think that, I guess that diet is important also, because you can have issues, you can be really good in processing cholesterol or have some issues, for example, and so, but the amount of cholesterol that you're assuming, or general health, like how fit you are. The brain is kind of like a muscle. They say to use it or lose it. So even if you have a predisposition to kind of like process well these proteins, but if you don't work, if you don't use it correctly as sport activities or also food uptake, I guess is important.
Francesco?
The answer.
So, about the diet, Leah actually covered most of it. Basically, yeah, also for cardiovascular reasons, we know that plaque formation is also a big confounder of any pathology, comprising also FPF that I study. And this plaque formation is not completely understood to this day. And actually, it's involved also in the plaque formation apolipoprotein E and A, actually. Yeah. So these can be increased by the wrong diet. In fact, as I said during my presentation, it's this high-fat diet with fat, salt, and fructose. It's also called Western diet for a reason, because it's very common in the Western society when we have this different lifestyle. So to answer your question, yes, lifestyle is so important for most of the pathologies. And may I ask the question?
I just want to make it clear that, even if you have some kind of genetic predisposition, eating well and doing physical activity is always important, okay?
Yes, the genetic predisposition here, it's called sporadic, so there is so many questions, so many people that have the predisposition but never have the disease. So the environmental factor and there the diet came in place. I mean, there are so many things that we don't know yet.
Just to address your question about diet, a couple of years ago, the Lancet Standing Commission published a paper, and they identified the risk factors which were beyond genetics that were related primarily to lifestyle. And one of the things they saw is that the combination of addressing poor diet, obesity, a sort of sedentary lifestyle and all of that would contribute to reducing the risk of dementia of 45%. So we're known for a long time that having a Mediterranean diet reduces risk for Alzheimer's disease. I mean, there have been studies at the epidemiological level that have confirmed that there is a huge difference between North and Southern Europe, which is related to the risk that having an unhealthy diet contributes. And therefore, the advice has always been a Mediterranean diet is much better than the Western, as you call it, diet, rich in, saturated fats and all of that. And then if I can make a comment about the, having a heterogeneous degree, I'm actually one of those who has probably also had a very heterogeneous degree, and I think is of added value, especially when physics meets medicine. We know that many advances in medicine have been related to the contribution of physics. The first MRI, whole body MRI, was actually created by physicists within Department of Medicine, where their knowledge actually meets the needs of medicine.
If I can add before passing to the next lecture, on the 19th of May, we're giving the honorary degree to Frank Hu, who is the director of the Department of Nutrition of Harvard University, and he was the one behind the first description of the effect of the quality of fats on cardiovascular health. So it was the one behind, is the reason, he and his team is the reason why we've basically taken out trans fats from pretty much everywhere, and that we take so much care about saturated fats. And his last publication, one of his last, he published like a gazillion papers. It's obviously, epidemiologists, how they work. But the last one is on GLP-1, and he proves that even if you are using GLP-1, if you don't care about your diet, even after you stop using it, but even at the same time, the risk factors go up again. So you can't really forget the diet. And it's, of course, as you say, I'm a little biased here. Thank you very much.